I'll turn it over to Natasha from Rada's team. Again, change of gears a little bit. You saw this morning more of an empirical analysis of three phase 3 studies. Natasha's going to share with us some of a more mechanistic approach to trying to understand TB disease progression. So take it away.
Thanks. Hi, so as mentioned, I'll be giving us an update on systems pharmacology approaches used by the Savage Lab at UCSF. We've been about 6 people that have worked on this project and components of it. I'll be acknowledging them as we go along in the summary. So getting right into it, when we look at our experiments, we know that we are often looking at our regimen, host, and bug in isolation. We know that these 3 components have a much more dynamic interaction, and this is where systems pharmacology has the benefit of looking at these relationships of PK, tox, resistance, and also our immune function that's being added onto our triage. So one of the issues why this is so important is that if we look at our current limitations in measuring treatment failure versus cure, we are limited by our assays. So we can compare 2 regimens, but we see that after some time we'll have some regimens failing, and we don't know the reason for this. So using modeling techniques or emerging biomarker data, we can fill in this gap of knowledge and gain depth in our understanding of how our disease is actually progressing and how our treatment is working. So to simplify how I'll be discussing the models today, it's always important when you think about modeling to think about your inputs. This is the data that we're entering. It can be raw data such as concentration over time. It can be parameters from literature. And this enters into our model, which is a set of equations. I'll just be going over the structural model for today. We usually also have error models, and this describes our population variability. And finally, we have our outputs, and this is our population profiles, so that we see our outcomes of treatment and also how the disease is behaving. So for building a systems model, we've sort of tracked our natural disease progression. So starting with our bug infection, our MTB infection, we then layer our host immune response as it would occur naturally, and then finally with this foundation, we can add our treatment response. So here's a structural model of our TB infection model. So we start with our infection in the lung. This is the bacteria, we program that the bacteria can move intracellularly and extracellularly. We also have that, it's a bit unclear there, but the bacteria undergoes natural growth. It is naturally cleared. And then we also have our natural mutation rates, which is the spontaneous resistance that occurs without drug to resistant strains. For this set of models, we've looked at mouse models and clinical data to fill in our TB growth and resistance. And then on the right-hand side, you can see the natural growth, intracellularly, extracellularly, and also spontaneous resistance occurring without any drug on board. So then we can start adding our host immune system. So here our infection is triggering an immune reaction. This causes activation in the lymph nodes and finally macrophage activation. I'm not going into too many details, but ultimately we have lesion formation where the bug is also present. So here, importantly, we also have a dynamic effect. Our immune system is killing bug. It's eliminating the bacteria, as well as the bacteria is evading the immune system. And we capture both of these in our model. This is specifically adapted from Marino and Kirshner, and that's where we got our model inputs. And then on the right-hand side, you see an output of the immune functions over time. So then finally, we can add our drug to this foundation that we've built. Here, we've included lung penetration, intracellular and extracellular compartments. We've also looked at our lesions, intracellular and extracellular penetration of drugs. And this becomes more interesting when we consider multi-drug therapies, where we can see that some drugs are reaching our lesion more, and other drugs are maybe staying intracellularly. From the PD perspective, we're looking at bacteriostatic and bactericidal effects. We're looking at EC50s, mainly from the hollow fiber model system. And we're also looking at drug pressure-related mutation and resistance at this stage. So this is a snapshot of the PK. This is generated from clinical data, and also our in vitro and animal models are aiding in understanding our intramacrophage accumulation. So here in this example, you can see in green, rifampicin, it's very highly concentrated in an intracellular space, and then drugs like pyrazinamide are more able to penetrate our granulomas. So Sneha Gupta from our lab has expanded on this model. So using MALDI MS data from the Rutgers lab, they looked at 5 drug concentrations within specific lesions. There were 8 lesions classified in total. These included things such as cavity, caseum from granuloma, necrotic nodules. And she was able to model this data looking at rates of penetration into these specific lesions and also the ratio at which they penetrate. And then on the right-hand side, we have some simulations where we see that at the top there, those drugs are actively reaching their target concentration, but drugs such as kanamycin are below our target concentrations in our lesions. So at the end, after laying all these effects, we have our full quantitative systems pharmacology model. And it's all about bringing together our host immune system, our mycobacterial infection, and also our drug on board. So this is the fun bit where we get to start exploring drug therapies. So our standard therapy is shown in white. So this is just a view of the percentage of the population cured on the y-axis versus time after the treatment starts. And we can see that if we play around with things such as dosage, duration of the treatment, and also reduced frequency, a pattern emerges where increasing our dose is very beneficial, and starting our treatments is also beneficial. And then comparing, similar to what Radha's mentioned this morning, if we look at low adherence in patients and low immune system, we see that these are real risk factors, especially low adherence, with less patients reaching a positive outcome and also taking a longer time for them to reach that outcome. So when we compare our simulated data versus clinical trials, here we have percentage of unfavorable outcomes on 2 factors. So this is treatment duration, looking at 4 months, 6 months, and 9 months. We see that our simulated data mimics the same trend from our clinical trials. And also looking at reduced frequency, we have the same pattern emerging from our simulations. The next step, of course, is to move from simulations to estimation. So far these data we've been using is sourced from literature, and we want to start estimating from a single model where we know that external factors are, aren't an issue in our modeling. And that means preclinical translation, because we know that the mouse model is still very useful to this work. So of course, mice and humans are not the same. They are scaling limitations. But when you start to really probe the mechanistic model, you delve deeper under the surface and you get closer approximations of what's happening on your translational research. So the next step for us is to simplify this model so that we have accurate assays that we can perform in mice. And this is a really important outcome for us, to have a final toolbox where we know that the assays we need to perform in mice to have a clear understanding of how a drug will perform clinically is the ultimate goal. So this is work that Imke Bartling from our group has done. So working with mouse data from Eric Nuremberg and the Johns Hopkins team. He's presenting later today, I believe. They looked at immunocompetent and immunodeficient mice, and also looked at different drug treatment strategies. And she was able to model this in the mice and then translate both the PK and PD to human data. And what's important here is she also had an immune system on board that I haven't shown you, and also the basic bacterial growth and disease pathology. So on the right-hand side, the green dots represent clinical trial data, and then her simulated data is represented by the black error bars. And again, you can see that this translation actually mimics trial data pretty well. So this is what we've implemented so far for our systems pharmacology model. We've looked at 7 known TB drugs with the ability to add more later. Our host, we've already included HIV positive and negative, but we'd like to consider how to treat patients, as mentioned this morning. For our infection, we are currently just looking at monoresistance, but we have the potential to move into MDR and XDR, and also strain diversity. Of course, the next step is to make this tool available to drug developers so other people can use it. So with the help from Ron Kaiser, who's a previous member from our lab, we've developed a web-based app that should be released in the near future. All of the plots that you've seen me present here today were generated from the app. This is the PK data that I've presented earlier. And with this app, you'll be able to change your treatment strategies, look at different regimens, and also look at different parameters and change those depending on your population. One of the outcomes would be population-based and seeing how much of your population is successfully treated. And with that, I'd just like to acknowledge our academic collaborators, our industry partners, and of course, our funding. And thank you to CPATH for having us here. Thanks.